Assalamu alaikum ji.
Walaikum
Can you
assalam,
people?
sir.
Uh, I'm sure biology classes. We had an introduction session on last Friday. I guess Huzaifa was there, and let's begin with our first lesson today. So let me share the screen. Can you people hear me clearly?
Yes, sir.
And can you see the screen?
Yes, sir.
Great, great. So let me get to the start. We discussed this molecules, enzymes. This is mitotic cell division, transport in plants, transport in human, gases exchange, infection. Uh, uh, we have some difficulty in internet in our area today, transferred me. So maybe we experience some difficulty or discontinuity during our lesson. I have been switched over to another internet. So let's see if there is no issue later in the class. Okay. So this is our first topic today, preparation of a temporary microscopic slide. We have two types of microscopic slides. One is microscopic slides. One is the permanent slide. And the other is temporary slide. So preparation of permanent microscopic slide is not in our syllabus. Whereas the preparation of temporary microscopic slide is in our syllabus. Okay, so we have this set of glass slides provided in our lab. We may have, these cover slips or cover glasses that we may be provided, and we may be provided some plant material or an animal material to prepare the temporary microscopic slides. Preparation of temporary microscopic slides involves around 4 or 5 steps that we'll discuss one by one. So let's see step number 1. So you see, this is a clean microscopic glass slide. So you can see this is few millimeter thick, and this clean glass slide is taken. So this is step number 1. Take a clean glass slide. Okay. And then obtain a plant material that has to be viewed under a light microscope, or the slide has to be prepared. In most of the cases, we are given an onion to peel the inner epidermis. You can see this is a piece of an onion. And this is, you can see this is a very, very thin, transparent membrane, which is known as an inner epidermal peel. Inner epidermal peel. So this has to be removed. This can be removed with this forceps. So second step is to remove. This is second step. Remove onion epidermis. It may be, inner epidermis, or it may be an outer epidermis, but generally it is an inner epidermis. So we can pull it with the help of a forceps and then place it on a glass slide. So this is where we have removed this thin membrane from this onion epidermis, or from this piece of an onion, and we have placed it onto the glass slide. So this is being placed on the glass slide, and then what we do is to place a drop of, next step is to place a drop of a dye, or flood this plant material with, these are the onion pieces. We have removed an epidermis from here and placed in on the glass slide, and then pour. This step is pour a stain or a dye on the plant material. Generally, there are 2 types of dyes used. It may be number 1, methylene blue, which is used for an animal materials, and the other is iodine solution, which is used for plant material. So this is an iodine solution which is being used, or which is being flooded on, the plant material here. So we place an iodine solution onto this glass slide. This is step number 5. Then a cover glass. This thin sheet is, this is a rectangular, very thin sheet, which is held in between your 2 fingers, and this is inclined on this plant material and is placed on the plant material. It is placed on a plant material in such a way that air bubbles are not trapped in between it. So it is first inclined. One edge is inclined here. So this is one edge is inclined on the slide, and then other edge is gradually lowered down. It may be lowered down with your fingers, or sometimes a needle is used. This cover glass is supported on a needle, and this needle may be lowered down like this. So this may be a needle, and this cover glass has been supported on this needle, and this may be gradually lowered down. The other edge is lowered down, so this prevents an air bubble trapped in between it. So this is how uh, a cover glass is placed on it. After we have placed a cover glass on it, the excess dye may be soaked with the help of a, so this is step number 6. Remove an excess, remove excess dye from the slide with a soaking paper, or maybe a filter paper, or maybe simply a blotting paper or a tissue paper. So this is how your clean glass slide is prepared for viewing under a light microscope. So this is the first step that involves preparation of a temporary microscopic slide that involves 6 steps. So this is, if we just review that one by one. So let's see. This is first step. Take a clean glass slide. Then the second step is, to remove this onion epidermal peel. Place it on the glass slide here. Then pour few drops of a dye onto it here. And then place a cover glass onto it. And the excess stain, maybe an iodine solution, or maybe, the methylene blue, whatever the dye you have used here. This excess is soaked with this soaking paper or with this blotting paper, and our clean glass slide is prepared. Now this is ready to be viewed under a light microscope. Before we go to the use of this light microscope, we should understand the different parts of a light microscope. Though we will not have any questions on, the role of the different components or the different parts of a microscope, but before we are able to use this light microscope, we should be familiar with the different parts of a microscope. So let's see, we discuss one by one. Starting from here, this is pair of eyepieces. These are used to view through these eyepieces the specimen which has been placed here. The specimen is placed here on the stage. So this is a stage. Okay. And this is a lamp here. We place a glass slide here, which is held with the help of these clamps. So there are clamps to hold a slide. And then there are different powers that we can change, known as objective lenses. Okay. We have this switch which turns on and turns on off the light, turns on and turns off the light, on and off the light. This is where we can increase or decrease the light intensity. We have the focusing knobs here. There are 2 focusing knobs here. One is coarse adjustment knob, and the other is fine focusing knob. Here we have the base, and we have a body here. So these are the different parts that you should be familiar with. We'll talk about some more parts that are used to adjust the light intensity, the condenser, and the stage movement knobs. So we'll discuss some more parts of it while we discuss the use of light microscope. So before we discuss the use of light microscope, let's see. I have this small video. It is of around 32 seconds. So 40 seconds video. So watch it carefully, and after this ends, we'll move to the other part of this slide, and we'll discuss the steps one by one, which would help to understand the use of a light microscope. So let's see for 46 seconds. So that's it. We will discuss these different steps one by one. So maybe we have a different order as it has been given in shown in this video, but we'll discuss all of these steps that are used to focus a specimen under this light microscope and view the different specimen under a light microscope. So let's see, this is the first step. You can see these are the objective lenses. These objective lenses have different powers. The power is written on each of them. This yellow represents 10x written on it. This blue represents this 40x written on it. And this white has 100x. And you can see there is another objective lens, which is on the other side, not visible from here. It has this, the red coding on it. And this is the smallest one, which is used for scanning purposes. And this has written an X4 on it, which is used for scanning purposes. We have this other objective, which is X10 or 10X. It is low power. And we have this X40, which is high power. And we have this other one, this here, which is 100x. These two, this 100x and this X4 is not in our syllabus. We have these two objective powers in our syllabus. This is low power and this is high power. So we can rotate this part. This is known as nose piece. We can rotate this clockwise or anti-clockwise, and we can adjust the power of an objective lens. In the beginning, we adjust the lowest power, which is an X4, if we are provided with that, right above, this condenser lens. This is a condenser. This is a condenser which focuses light onto the objective lens. Okay. And then we have, this is the other view, which shows this lowest power X4 that has been adjusted right above this condenser, and